Here's the last video lecture for this week, October 26th, Friday. And we've been looking at these different topics. We looked at cell division, and we saw these chromosomes being divided up into new cells. And we looked at inheritance, and we saw the basis for inheritance being the chromosomes and genes that come along with those. Well, it's fine for cells to have the chromosomes duplicated and be donated to new cells. It's fine for a parent to give genes to their offspring, but what do these genes do? Our focus now is looking at these genes being used to do something, and that something is to make a protein. When you've got certain genes, then you can make certain proteins, and you can carry out certain functions. So that's the focus of this, and what we started looking at was this relationship between DNA, where you've got a sequence of nucleotides that's used to make a sequence of nucleotides, and RNA, which is similar, made up of the same type of molecules, which are nucleotides, a sugar and a phosphate and a base. And you've got amino acids is what you're after, which makes up proteins. So let's look at this idea of codons. Why are there 3 bases in a codon, or 3 nucleotides in a codon? Well, it gets down to the goal, the information that you need to convey from RNA to proteins is making this transition from a chain of nucleotides to a chain of amino acids. So how many different nucleotides are there in RNA? How many different types of RNA nucleotides are there? There's only 4. There's a sugar and a phosphate and a base where the base is an A. That's 1. There's a sugar and a phosphate and a base where the base is U. That's 2. There's a sugar and a phosphate and base where there's a C and one where there's a G. So there's 4 different types of ribonucleotides, each one that has a different base. So the goal is you've got some message in RNA, the message being the cell wants to know, what amino acid do I bring in? So you've got to have a relationship between this nucleotide and one of the amino acids, telling the cell which amino acid goes where. So a codon, you think about a codon has 3 nucleotides. It consists of 3 nucleotides, and so does an anticodon. So the question that we're asking here is, why is that? Well, if one nucleotide corresponded to 1 amino acid, then you could only code for 4 different amino acids. A would correspond to 1 amino acid, and U would correspond to another amino acid, and C another one, and G another one. There'd only be 4 amino acids that a codon of 1 nucleotide could carry out. And how many naturally occurring amino acids are there? There are 20. So, in other words, with these 4 letters, you've got to be able to spell 20 different things. If you just use these one at a time, that only spells 4 things. Well, what about using 2 of these at a time? Having a codon that's AU, and another codon that's AA, and a codon that's UU, and a codon that's CC, and a codon that's GG, and a codon that's AG, and CG, and UG, and if you added all those possibilities up 2 at a time, you would discover that there's 16 different combinations for these 4 letters, 2 at a time. So if a codon consisted of 2 nucleotides, AU was a certain amino acid, and AA was a different amino acid, and UU was a different amino acid, you could code for quite a few different amino acids, but only 16. So 2 at a time is not enough to get to 20. 2 at a time would allow you to code for 16 different amino acids. If you have 3 at a time in a codon, the codon is 3 nucleotides. So the information coming from RNA to transfer RNA is 3 at a time. Then you can code for 64 different amino acids. You could code for 64 different amino acids. What that means is there's 64 combinations of these 4 letters, 3 at a time. So there would be AAA, that's 1. There'd be AAU, that's 2. There'd be AAC, that's 3. There'd be AAG, that's 4. You could have AUC, AUG, UCG, UUC, UUG, CCG, CCC, GGG. Anyway, if you went through all the different possibilities, you'd find out there were 64 different possibilities. That's more than enough. 2 at a time is not enough, only 16. 3 at a time in a codon is enough to code for 64 different amino acids. That's 3 times as many as you need, but you need at least 20. So that's why a codon has 3 nucleotides. So does the anticodon, because you need that many possibilities to correspond to at least 20 different amino acids. In fact, there's a lot of redundancy. So here's a table that's put together. We know all this because people have investigated this and they've discovered what sequence of nucleotides in RNA corresponds to which amino acid. So these letters, 3 at a time, UUU, UUC, those are letters in a codon of messenger RNA. If the 3 nucleotides in a codon are UUU, then the transfer RNA that's bringing in an amino acid is going to bring in this amino acid, phenylalanine. If the combination of those 3 nucleotides is UCU, then the amino acid that transfer RNA is bringing in is serine. But there's some other possibilities for serine. If the transfer RNA is matching up with a codon of UCC or UCA or UCG, all 4 of those, there's a transfer RNA that has attached to it the amino acid serine. So you can have 4 different codons in a messenger RNA that are going to result in the same amino acid being brought in. However, this sequence of nucleotides, UCU, is never going to code for any other amino acid besides serine. So you can see that for a number of these, there's 2 different codons that correspond to phenylalanine. There's 2 different codons that correspond to leucine. And there's 4 more down here. So there's 6 altogether that correspond to leucine. And you see some other ones where there's 4 that correspond to proline and serine and threosine. And there's a couple for arginine and glycine. These are the 20 different amino acids. And these are the 64 different codons, the 64 different possible combinations that you get when you use those 4 bases 3 at a time. So there is redundancy here, but you still have the situation where whatever codon is in the messenger RNA, ACU, that's very specific for no other amino acids except for threonine. ACU, it always is asking for a threonine to be brought in at that particular part of the synthesis of that polypeptide chain. Well, if you look at it more closely, there's this repetition, like I said, this GUC, the only thing that it tells a cell, if that's the codon, is to bring in this amino acid valine. This one, UGU, the only information that that codon contains is to bring in, for the transfer RNA, to bring in cysteine. Same thing here, AGU. The amino acid that goes right there, nowhere else, is serine. So that's how there's redundancy in that you can have 4, 5, or 6 different combinations of those nucleotides that code for the same amino acid, but one codon only codes for one amino acid. One amino acid can be coded for by a number of different codons. So if that doesn't make sense to you, then stop and think about it for a minute. Now, another thing is that I mentioned before is that the ribosomes are going along the messenger RNA, they get to some codons where there's no corresponding transfer RNA. There's no transfer RNA out there that matches up with its anticodon. There doesn't have an anticodon that matches up with that codon. Those are the stop codons. So there's a sequence of 3 nucleotides in a messenger RNA. It's not a signal for any particular amino acid, but it's a signal, nevertheless, for that process of joining these amino acids together to stop. So in other words, the gene is done. You're done expressing this gene. You're done putting the amino acids together. That's where the process stops, and now you've got 141 amino acids, or you've got 146 in the order that they're supposed to go to make that protein. And another thing, we haven't looked at it so much, but if you looked at these figures earlier on, you might have noticed that this codon, AUG, is at the start of every single gene. This codon that signals bringing in this amino acid methionine is how every single gene starts. Every single messenger RNA starts with this codon, and this amino acid methionine is the first one brought in. So that's a signal to the cell. This is where you start this process. One amino acid methionine, and then one of these other 21s, and another one, 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 until you get to the very end. In that hemoglobin example, it might be 141 amino acids, all assembled, and then you get to one of these 3. If the codon is UAA or UAG or UGA, there's no corresponding transfer RNA. The whole process stops. The chain of polypeptides comes off of the ribosome and goes on its way to become a functional protein. Okay, so now we've looked at this whole process of starting with DNA, making RNA, messenger RNA, using transfer RNA and ribosomal RNA for this process of translation to make a protein based on information in RNA. So you can take information in DNA, in genes, on chromosomes that are divided up during mitosis and meiosis, that are passed on to new generations in inheritance, heredity, and actually use that information to make a protein, which is something very functional, very important for the success of all these different organisms. You've got to use these proteins and express these proteins to, or express these genes to make proteins if you're going to survive. These proteins, we've got thousands of different proteins based on thousands of different genes, and those thousands of different proteins carry out thousands of different functions. Some structural, a lot of enzymes, a huge number of enzymes. So this is really important, that you've got information in DNA that's used to make proteins. It's sometimes called the central dogma of DNA in or gene expression in biology.